Our next presenter is a professor in the Department of Biology at SUNY ESF and is the director of the Thousand Island Biological Station right here at Governor's Island. He's also past president of Save the River Board that he served on for 22 years. That's when I first met him. Please welcome John Farrell to discuss the health, habitat, and stressors of the pike family. I'm going to say it, Asocidae, including northern pike, muskellunge, and grass pickerel.
Hello, thank you everyone for that warm applause. So let me make sure everything is going to work here. There's my pointer. This is kind of intimidating. You guys you didn't warn me about this, but anyhow, it's what's that?
It's for the live stream.
Yeah, right. Oh, live stream, I didn't know. I better be careful what I say. But anyhow, it's wonderful to be back here at a winter weekend. I think I was here and I gave one of my first presentations at this venue, maybe at the first winter weekend in 1990. I found a picture of that. I decided not to show it today, but but anyhow, it's great to see so many friends and supporters of this magnificent place that we all enjoy. And I'm going to move right into the talk here. So, as Bridget mentioned, we've developed this amazing place on Governor's Island. So the Lewis family donated this island to ESF back in 1973. Administrators, see, I'm going to get myself in trouble. Administrators kind of played there. The president built a dock at the island. He kept his sailboat there, and it was used kind of as a administrator retreat. But then some people took over the facility, biologists and some chemists worked there from the college. Some of the early winter navigation studies, folks used the facility at that time. And then Bob Werner, my mentor and professor at the college, worked with Steve LePan, and then I came on and we started a program to look at this mysterious decline of the muskie back in the early 1980s. And then I took the facility over after Dr. Werner's retirement and helped build the Thousand Islands Biological Station with so many partners and people. There's literally, I think, 300 alumni that have come through the program. You think you've probably seen all those graduate students and undergrads pulling seines and collecting data and monitoring the river over these past few decades. And it's really it's not a job that one person does. It's like a massive team. So I'm very privileged to have built a career around the river, a place I love, a place I grew up on, and I'm still living the dream here. So we do this is going to make sense to you, but we kind of designed this program revolving around the river's ecosystem. And at the base is fisheries management, making fisheries management decisions. And we really study like a lot of other aspects of the river, but this is where the money comes from. So people are very interested. The fishery is a big reason why we're here. I'll get to that in a moment. But we study population and food web ecology with both fishes and vertebrates, and we've really expanded into a lot of work into our wetlands, which are the basis for reproduction and much of the biodiversity that we find in the river. And we also have gotten into invasive species ecology, and that goes without saying. And more recently, we've been involved in restoration ecology, and you're going to hear more about that. So it's been a pretty exciting ride. Many of my colleagues work all around the world studying these amazing places, the Galapagos, Palau, all over the world, studying ecology, and I've stayed in one place, and it has not been boring for 1 minute. So we have an aquatic monitoring program I mentioned, and we get a lot of our funding and support from DEC and also from U.S. Fish and Wildlife Service, but also other grants. And we monitor a lot of things like temperature, water level, dissolved oxygen, arrays of sensors that are out in the river that are maintained. We do limnological surveying, nearshore wetland vegetation, juvenile wetland fish and invertebrates, nearshore fish community and aquatic habitat, and nearshore adult fishes. So it's a pretty significant task just to collect these baseline data, and it's kind of like analogous to when we go to the doctor and they take your blood pressure. We have a record of a lot of things on the river, and you know, collectively, this place is pretty well served by people who care about the environment. So it's how'd that Bills thing get on there? Hold on. What happened there? Well, yeah, I am a Bills fan, but they haven't contributed financially to us yet. Maybe Bills Mafia will pick this up. I don't know. But anyhow, so the point being that we just have an amazing set of collaborators. You know, two big ones here. But also a lot of other organizations that make all this work happen. So today, with that little bit of introduction, we're going to talk a little bit about the recreational fishery, what I've called the challenge, and how the rivers, everybody recognizes the changes in the river, and that presents a challenge to the native species. We're going to look at some of the trends in muskie and pike, what's being done about it, talk about a new kid on the block. I'll let that pique your interest, and then what can you help? So that's kind of my goal here today, to go through that. So the fishery is massively important ecologically. You think, you know, Lake Ontario, the predator base fell apart in the 1960s and early 1970s, and there were piles of rotting invasive alewives on the beaches. Beaches were closed, water quality was horrible. And part of that is because of the loss of the top-down effects of a predator. We have native predators here that maintain ecosystem balance, and it's a service that maybe we take for granted. But, you know, in terms of the fishery, it's also a huge economic importance. It always has been to riverfront communities. And this was a study that was done fairly recently, and it was done during the flood. So I don't really trust these numbers because I think more people are fishing now than they are at this time. But, you know, the total economic output of the St. Lawrence fishery was valued at $84 million, direct expenditures of $28.4 million. So here's a link here. So it's just an incredibly valuable fishery. This is like right before one of the tournaments, or during one of the bass tournaments, and you know we've got the bullseye being ranked as the #1 bass fishery in North America, and we're not talking about that today, but I'd be happy to come back and talk about that. You know, other challenges are it's the St. Lawrence Seaway, right? And this is the existence of Save the River revolves around this fact, and you can look. This is actually the this heat diagram shows the transits of oceanic shipping, and there's one transit that goes right across to Europe and the Baltic region. And you know, so there's this Great Lakes oceanic, transoceanic shipping. About 14% of traffic here is from the transoceanic shipping. And we've had a lot of invasive species. I mean, this place, when I started working here, there were no zebra mussels in the river. It was a very different place. We worried about exploitation effects on fisheries. We didn't worry about habitat. We didn't worry about invasive species. So all these things have been really recent. I don't feel that old, but it's like, so invasive species have come in. And we're not going to focus on that today, but there's one big one here that we will talk somewhat about, which is the round goby. So about 100, I think there's 190, maybe a few more invasive species that have come into the system. They say roughly about 2/3 of those are associated with the seaway. So there's this concept that famous ecologist Dan Simleroff, who I got to meet once, who came to ESF, came up with this concept of invasional meltdown, where synergistic interactions among invaders may lead to accelerated impacts on native ecosystems. So we certainly see these sorts of processes operating here in the St. Lawrence River. So it's changed the place quite a bit. So, you know, this is, I thank Spencer Butzler for taking this photo last fall. So here we are in western Goose Bay, Cranberry Creek, and you can see this typha. This is hybrid cattail monoculture. So this is all an invasive species that's really altered our wetlands. It dies in place, it grows and dies in place every year, and it's causing like really rapid accelerated peat accumulation in our wetlands. And this, you can see how low the water was this past fall. So these are like really large mud flats. So that's all like decomposed sediment year after year that builds up layers and really affects like the ability of fish and other organisms to access the floodplain, which is a big issue that we have here that's related to water level management. We've also seen, because of that typha, here's Flynn Bay, there's a ubiquitous effect of this typha. All the wetlands, there's four major geomorphic types that all have the same impact of water levels, which maintain the vegetation, diversity, and zonation in wetlands. And they've all been invaded by this typha exoglauca, this hybrid cattail. It's a little hard to see this, but this is dissolved oxygen levels in the St. Lawrence River. This is at Governor's Island. You could say they stay nice and high. They do decline, so there's processes where organisms, bacteria, and other things can consume oxygen that drive the oxygen levels down. It's called a heterotrophic process. But in the wetlands, there's like lots of oxygen early, and then it's flatlines, and it creates what we call anoxia. So there's certain organisms that are really challenged to exist in wetlands following spring, late spring, and then early summer during this anoxic period where there's certain fish that can gulp air and stuff like that, but other fish have to leave these important nurseries because of low oxygen. So we study that, so that's a big effect. Another thing is just loving the place to death, right? We're seeing massive development in this region, especially after the floods of 2017 and 19. There's a lot of interest in the river. It's wonderful. People love it, but there's a downside associated with that, was how it affects our nearshore aquatic habitat. So there's many other issues. I could sit here and talk to you. We could have like a symposium for days just talking about the issues facing native fishes and other aspects of our biodiversity here in the river, but I don't have time for that. But I do want to focus on a really special organism that we have here, which is the St. Lawrence muskie. The river is well known for its spotted form of the Great Lakes muskellunge, Essex masquinonge. Incredible organism, grows. We've seen them up over 1.5 meters. The biggest one in our angler data set was 61 inches. And you hear about the wonderful catches that people get in the river every year. So it's just really known for the immense size of this animal, but it's also a very sensitive animal. They live to about 30 years. They spawn, and we've done a lot of research on them, and they spawn in the same nursery sites year after year. So they have a fidelity called philopatry to go back to the same spawning sites year after year. So they have this incredible life cycle that involves wetlands. So fortunately, there was a decline before, and Save the River was at the center of this. And it was primarily due to over-exploitation, but it led to the development of a management plan to perpetuate muskie here as a self-sustaining component of the fish community first, and a quality trophy fishery second. And it led to a lot of like incredible environmental development. We feel like we're leaders in muskie management across the range of the species, and we've seen a lot of work towards catch and release. There's under the Great Lakes Fisheries Commission, there's an ASACID working group. It used to be the muskie working group. So we have members from the US and Canada and other researchers that look at data and help guide priorities. All that monitoring is kind of born out of this critical habitat identification, and it's led to increases in size limits to recognize that they have to reach a big size to reproduce, and also catch and release, and that's where Save the River came in the early years in the development of the Muskie Release Award, which has really changed. Really, the credit goes to the anglers for recognizing the need to not keep every muskie they caught and come into Clayton with a white flag, but release the fish. So we have this wonderful progressive management and conservation history, and that We felt like rock stars. The population was coming back, and in the early 2000s, there was a magnificent period of fishing, and the populations were burgeoning. But we recently had this massive die-off. Not that recently. This is actually data that was collected 2005 to 2008. This photo was taken by Dr. John Castleman, who's a noted saucid biologist in Canada. And they have a dead fish box over on Georgina Island, and people would put fish in there, and it started filling up and overwhelming with dead muskies. And we started going out and finding dead muskies all over the river. This was 2005 to 2008. We recently published a book on muskie management, and I'm really proud that 3 out of these authors all came. This is an 800-page book or something like that. I might be misquoting myself there, but the 3 of the 5 authors came from the Thousand Islands Biological Station, so we're very proud. So we recently had Anna. She's presented here. She was my PhD student for 6 years. She completed her work, and she's studying the virus that caused that die-off. And she identified that this virus is evolving. You can see all these haws. These are new mutations that have happened to the virus. So the virus is continuing to change, is one of the take-home points. So we're seeing like an increase in the number of isolates, so that's kind of like evolution of the virus. And then when we look at the reservoir of the virus, it's pretty obvious that the round goby, an introduced species, is a driver of maintaining VHSV, viral hemorrhagic septicemia, which the muskie is extremely sensitive. There's like 29 known fishes that are impacted by VHSV, and I think muskie are ones that are extremely sensitive to it. We work closely with the Cornell Veterinary College in this study. So gobies are the key to VHSV being perpetuated in the ecosystem. So here's some more data from a chapter in that book. This fish actually I found with my brother. We were heading to Foxy's after working a long day and found a dead muskie in Blind Bay that had one of our tags in it. And you can see these are data from our spawning survey. I got Blind Bay up here on the top. Everybody's so concerned. We started standardized indexing Blind Bay back in 1990. We turned all our data over to the environmental consultants at CBP. And it's not just muskie data, there's thousands of data points. So that 53 species quote comes from the TIBS data set. So that the data speaks. And muskie populations, they were doing so well that we decided to stop the annual survey in 2000. And then we picked it back up in 2006. So that we only sampled 1 year because it was so much effort. We wanted to work on other things. When we started resuming the survey in 2006 till today, we've seen declines in the population. So Blind Bay has really lost a lot of its muskie. They're not gone. We've done eDNA studies that show these sites still have muskellunge, and now we see sporadic catches in Blind Bay, also swan. Flynn Bay's population survived, and they still reproduce and produce young there. When we do monitoring at 11 sites throughout the region, and you can see that gap when we didn't sample those years. You can see how many we caught in 2003, which was a record, and we've seen a decline. So there's more and more effort to find fewer and fewer fish in Thousand Islands muskie spawning grounds. So what are we doing about it? So I'm going to move fast through some of this. There was this thing called the Fish Habitat Conservation Strategy that's been developed through the relicensing of the power dam. I think you heard from Scott Schluter last year on some of the work with the hydroacoustic array. And Scott manages the FEMREF fund, Fish Enhancement, Mitigation, and Research Fund for the U.S. Fish and Wildlife Service. And there's a fishery advisory group with a lot of different people from DEC and other agencies that help determine if proposals get funded from that $26 million fund when it was originated. And we've applied and received a lot of those proposals and developed this thing called the Fish Habitat Conservation Strategy with our partners, including Tilt, Save the River was involved early on, and then other agencies that can do stuff. So we actually look for problems with reproductive habitat, plan and design. We work with groups that can implement things like Ducks Unlimited, USDA, Fish and Wildlife Service, DEC, and then we study whether they work or not. So there's a lot of restoration done where the evaluation part isn't completed, and sometimes it's kind of feel good, but you don't really know the outcome. So this is an adaptive management process that's been going on at Tibbs, and one part of that is the muskie recovery program. So we actually obtain brood stock from the river. The Lake St. Lawrence region near the power dam hasn't experienced the same level of decline as the Thousand Island region, so we're Fish and Wildlife Service nets down there. And we also get brood stock from here in the Thousand Islands. They're all from the St. Lawrence River, nowhere else. We're doing genetic studies to make sure we're very concerned about maintaining the genetic uniqueness of the population. So we take these brood stock, we collect eggs and fertilize them in our lab out on Governor's Island. And there's a couple of videos here. How do I get these to play? Play videos. There we go. Oh, so anyhow, these are some fingerlings from last fall that we raised out on Governor's that were released. This is a really cool one of a fingerling. The next one, if I can get it to go, please. There it goes. See that little muskie? It's just gonna dart off. So that's in Blind Bay. And so these fish are really behaving like wild fish. You have to tape the cooler shut. They jump out. They're absolutely insane. And you don't want to have a fish that you could walk up and tap on the head. So I'm pretty happy about this. So these are the restoration locations. So a lot of work was done to understand where muskie spawning and nursery occurs. I think you heard about Matt this morning. I think he talked a bit about some of the work that they're doing out of the St. Lawrence Institute. But we've identified something like 120 spawning areas throughout the entire international St. Lawrence River. A number of those, really the populations have been declining. So we're stocking these fish in hopes that they'll imprint on these sites and return. This is restoration. We do not want to be stocking muskie in the river. We want self-sustaining, and that is in our management goal. We're doing this to restore spawning populations. So how do we evaluate it? So since 1983, we've been seining in the river. So I'm showing you a piece of our seining data. We developed a series of 11 really important nursery sites that we've been monitoring since 1990. This is data from 1997 till last year. I didn't get time to add this year's data, but you can see all these fish here. This is for those of you who want to go seining, we always look for volunteers, by the way. It's quite a way to really learn a lot about the river. But anyhow, we drag this thing 30.5 meters, 100 feet, and we process all the fish that we catch, and we collect the habitat measurements. And we were catching, something like one or two. You can see these are incredible numbers of muskie on the nursery ground back in the early 2000s. These are all wild fish. And then VHS hit, you can see the population decline. And then you see a population to crash here, like 2015 to 2020. So we waited a while. This was a pilot study where we stocked muskie fry, and you can see this increase. So that was due to the fry stocking. And this is also due to fry stocking. So those increases are from us putting fish back into the nursery sites. The fingerling muskie, which are larger, go in the fall, so they're not represented in this distribution. So we're now, the fingerling fish get a pit tag. We've developed this thing called the Muskie Citizens Science Angler Program, and anglers have kits, and it's not this big. This is not to scale, so it's actually like this tiny little tag that we inject into the fish. They heal, we've tested that. And you can use this wand to see if that tag is in the fish, and we would know when it grows to adulthood that it came from the Thousand Islands from our program. So that's been going on since 2017. I think we've stocked something like 150 50,000 fry and 26,000 fingerlings in the river. The amount of work to do that is just, I don't even like thinking about it, but we've learned a lot about muskie along the way, and that's the evaluation. So another thing I wanted to speak about is this recent development. You heard from Scott about the array that's been installed in the river and throughout the lake called GLaDOS, the Great Lakes Acoustic Observing System. So there's listening devices. They're not listening for us, but they're listening for little beeps that come from tags we can put inside fish, and we can study their movements. We're collecting data right now as we speak on muskie out in the river. We have 13 muskie with these tags in them, and we're following up on studies that Steve LePan led, I was involved with, on muskie behavior, where we learned muskies were migratory, we learned about the spawning fidelity, and he used radio tags. Now they have these receiver arrays. They're all over the ocean. So this is being maintained and led by U.S. Fish and Wildlife Service to put all these receivers in. Look at all those receivers. Those guys go out and they lift those, and you have to download the data each year. They're throughout. There's other groups in GLaDOS that do the entire Great Lakes. They're up and down the East Coast monitoring sharks and other animals. So it's fascinating to have this technology to learn about muskie behavior. So we've been tagging fish in eastern Lake Ontario, here in the Clayton area when we can find them. We don't use fish that were used in the broodstock, so it's particularly challenging. We're also working with Canada, Collin Lake, out of the Glenora lab, and Bay of Quinte. They actually tagged a fish last year, so we have Canadian partners, and we work around the Waddington area and also around the dam that we've been focused on. We're trying to get 34 tags out, so this is just getting started. Here's John Paul LeBlanc. JP is one of our scientists. He's not here today, but he is really the leader of that program for us. He's doing a surgery. He cut his teeth in Georgian Bay, working on muskellunge conservation, and he's been with Tibbs now for 9 years as a postdoc and now a scientist. This is the little tiny incision that's done. It heals up. This fish was actually recaptured by an angler in the Racket River, and we found that it was healing. They're absorbable sutures. This is the tag. It's about the size of a 12-gauge shot shell. I think some of you know what that is. And so anyhow, that was the healed wound after just a matter of a month or something. And then here's some of the results. So it's incredible. The first year we had 38,000 detections. Now in 2024, we just downloaded, we have almost 300,000 muskie movement detections throughout the river. I mean, this is just like blows me away. So these are where the larger the circle, the more detections. So we're getting inferences about muskie ecology across the entire ecosystem with this study. Fascinating. Here's a couple of highlights. I could talk about this for days, but this is an animal. This was a male muskellunge that was tagged in eastern Lake Ontario in Mud Bay, and what we see is these upstream movements, and that's exactly what Steve saw. Is a lot of our fish, like post-spawning, move upstream. Mud Bay is a known spawning and nursery site where we monitor. It went out to Fox Island, another known spawning and nursery site, and then it moved further out in the lake, and we saw this upstream behavior. This is the granddaddy though, so watch this. So this is a male muskellunge that was tagged in Waddington. Please go. Huh. Oh, come on. It's kind of jerking around with me here. Hold on. There's that. We've seen that. Please play. There it goes. So those little dots, the red dots represent 2023, and you can see that fish moved all the way to Amherst Island, a trip of, it's a 320-kilometer round trip this fish made from its spawning site in Waddington out to Amherst Island in Lake Ontario. And it did this, and it stops at particular locations. The blue represents this past year, so we have 2 spawning cycles. Guess what? It did the exact same thing. So this fish is using if anybody wants a lesson and connectivity and the complexity and importance of the whole system, these fish demonstrate that. So this fish moved 640 kilometers straight line to complete 2 spawning cycles. It stops at Howe Island every year. That looks like a place a lot of the anglers know because this is near 40 acres and where people fish for muskellunge. They like Canada. They stay on the Canadian side. You guys read into that what you want, but but anyhow, just fascinating, big scale movements by these fish. So, and it's not just like, we have these depth tags, so the tags tell us what depth the fish is at that time. It also measures temperature, some of them. So we have a lot of data, and it's cool. You can see these fish, they make really rapid movements. They go to a place and hang around. There's a spot near Chippewa Point that's really important where a lot of fish hang around, and then they head to Waddington to spawn, and then they're going back. And you can see that they're using depth strata. So when they're spawning and pre-spawning, they're coming in shallow. When they're migrating, they're going deep. So this is going to take us, these are 10-year tags. We're going to get 10 years of information on a fish. Like I said, some muskies live 20, 30 years. So it's just fascinating data that's going to tell us a lot about the importance of habitat to these fish in the river. So moving on. Okay. I got too many animations. So we also tagged fish below the Robert Moses Saunders Power Dam, and they tell a very different story. It's playing right now. And they kind of pinball, and they don't have a really long or significant home range. So we're really interested in the influence of the dam on fish migration, and you can see that they're kind of residents staying in the same area. We did have a fish this spring that seemed to get through the dam and went upstream. So we're just like really perplexed how that happened because we have all the possible routes to get there covered with receivers, but that did happen this year, so stay tuned on that. So very different behavior below the dam. I have to switch to another of the family Asacidae, the northern pike that we all know and love. This is the littoral predator, bread and butter fish to a lot of anglers. A tremendous fishing history here. There we go. Oh, so you press it once and then so anyhow, very proud to say that we've done a lot of work with northern pike over the years. Anytime we studied muskies, we also studied pike because they're cousins. We just got the cover of with some research with partners at University of Victoria. This is G3, it's a genetics journal, and they studied northern pike, which crossed Beringia from Europe into Alaska and then spread across the North American continent. And something fascinating we discovered, actually there was a scientist, Jan Geigen, who first found it right when we were finding it, but the northern pike have lost their sex-determining system in all fishes east of the continental divide. So that was a pretty big finding that came out of this paper, and we don't know what's replaced it. So this is different than what you see in European pike. And what's interesting is we've seen a sex ratio shift in our spawning northern pike population that's between 60 and 70% female. So we're seeing like this feminization. You'll catch these little scrawny males. You look at this female full of eggs and you're like, okay, how are you gonna do that? So it's kind of very interesting. So it's leading to some new research. Agencies do hard work monitoring these populations. This is a compilation that was done out of a group of pike scientists in Canada, and they recently gave this poster. But what's interesting is here's the Thousand Islands here, Middle Corridor, Lake St. Lawrence, Lake St. Francis, completely different hydrology further downstream. All pike are like collapsing in terms of the catch rates that they're seeing in their gill net index. And so it's not just here, and it goes further downriver as well. So we're seeing some problems with pike. We've been studying those here, and we know at least like further downriver in like what's called Lac St. Pierre, they've completely converted the floodplain to agriculture. Here, I mentioned the typha has taken over. So northern pike used to run up rivulets and spawn along shorelines, and now they're being blocked by this cattail. They love flooded wet meadow. This is a wet meadow at Chippewa Creek. I think it's near Alan Newell's property. And pike used to be able to spawn in these areas, and now they've got this wall of cattail they can't get through. We've applied, as part of the fish habitat conservation strategy, working with Fish and Wildlife, doing angioplasty on our marshes, to use an apt analogy here. And the sediments, I think you may have heard this, the sediments that were spit out by this machine that creates these channels germinated native seeds. So these seeds have been buried by typha for 60 years, and then they germinate beautiful native sedges and other plants. So they were still viable. We've been monitoring these projects with this hoop netting, mini mesh hoop netting technique. And when we run our models of young of the year emigration from wetlands over time, spring water level, and habitat are the factors that explain the variation in this data set. So it shows that our water level management and the invasive species are regulating these recruitment processes of young pike. Okay, so and the other exciting thing about this is unmodified habitat that's still functioning, and the modified habitat are producing pretty equally. So we're able to manage and replicate reproductive habitat. Build it and they will come. This is so we've been doing this quite a while now. This is our data going back to 2011, and you can see these huge spikes of northern pike reproduction, 2014, 2017, the year of the flood. We had this huge pulse. Then it was really bad. Now it's recovering again. And look at how the unmodified matches the enhancements. There's a lot of enhanced habitat. It's something like 300 acres. Something 100 miles. I have to look up these numbers, but hundreds of miles of channels, 100 miles of channels, and many, many acres of potholing and removing of cattail to open habitat. A lot of work. DU, U.S. Fish and Wildlife Service, and we're monitoring the outcome. And so what's interesting is we're trying to connect this reproduction to the adult population. And that's taking more time because this huge year class, we caught thousands of young northern pike emigrating in marshes following the floods. And I thought, here comes our year class of adult pike finally, and they didn't show up. What happened to them? So it's kind of a mystery. And so we're really looking at the connection between, and these aren't like tiny pikes. Some of these that emigrate late in the emigration are quite large. So you would expect they would survive. So it's, that's science. Okay, the new kid in town. This is another assassin. It came from an introduction probably to Lake Erie, and now it's here. It's the chain pickerel, Essex Niger. Look at that, it's a beautiful fish. I caught this one in French Bay. Not a lot of people are aware of this, but here's a little video showing 2008. These were abundant in Black River Bay prior to this. It's giving me problems again, sorry. Go. No? See how is that an insect or my cursor down there? Huh. I'm pressing the right button. There it goes. Okay, so this is the expansion of chain pickerel. So back in 2011 was the first known occurrence of chain pickerel in the province of Ontario. And now they're spreading through the system, and they're moving downriver. If you want to catch these, they live in shallow water. So the other pike and muskie, they come into the wetlands and they leave after they spawn. Chain pickerel stay. So I caught one in Aunt Jane's Bay last summer that was 21 inches, and I caught it in 18 inches of water in August. Okay, so they're staying in very shallow water. So they're having like a different sort of an effect. And here's some of the data that we've got on chain pickerel. This is the number of sites. This is chain pickerel abundance in the Thousand Islands, and you can see we'd see them. Oh my gosh, we got a chain pickerel. And now they're like growing, and there's populations developing. So these are adult chain pickerel. We're getting them frequently in our nets. They're hybridizing with northern pike. And here's sites. So you can see here's Blind Bay. They're really taking off in Blind Bay and other sites. So we're getting a growing chain pickerel population. JP calls this the fish of the future. Like it or not, it's going to be here. Chain pickerel catch per unit effort. This is our index trap netting. So we're seeing this dramatic index. Who's catching chain pickerel? Anybody? Anybody yell out that are getting them? See, nobody's catching them, but our students are. They fish for them and they do very well. It's because they're in these nearshore habitats. It's kind of like fishing for largemouth. But this is what we're looking at. It's like, what impact is this going to have on our other saucids? So this is our seining index going back to 2002 for northern pike, and you can see this precipitous decline in the production of young in nearshore habitats. And here's chain pickerel increasing. So they're, inverse lines. So chain pickerel, young of the year, are right there. They look a lot like pike, but they're different, and they're becoming the fish that's going to be abundance. I don't want to leave out this guy, our lovely native Esox americanus vermiculatus, which is the grass pickerel. Most people don't know we have this incredible fish that lives in wetlands. They're quite abundant in creeks and wetlands around here. This is one of the biggest ones I've ever seen. So they're not really part of the fishery, but they're part of the ecosystem. They're a species of concern in Ontario. So, so anyhow, how do we manage fish? So we've got, Ontario has these fishery management zones. New York has a fishery management zone. We're here in zone 6, region 6, and here's the regulations. Pike 22 inches, 3 fish. Muskie 54 inches, 1 fish. Most people let them go anyhow. But some work was done. Jana Lantry is the fish manager here. She's very active and leads our programs administratively and is super supportive. And Les Resegui is the St. Lawrence River biologist, and he manages, helps manage the fishery and collect data. 22 inches, it might be too small. We're seeing fewer larger pike. We've reduced the bag limit from 5 to 3. The guides really accepted that. They have a very different system in Canada where you can take any size, and you can take 6 for pike, and then they have a conservation license where you can take 2. We have common regulations for muskie on both sides. So these are the levers. The other lever of changing things is what we do with the abbreviated review. So we have a seat, and Tom Brown's here. He's part of the St. Lawrence River Board, and I'm sitting on this Great Lakes Adaptive Management Ecosystems Committee, and we're developing indicators for how the hydrology and deviations from Plan 2014 are going to affect fish reproduction. There's an amazingly talented group. So this abbreviated review is going on, and it's optimistic that hopefully we can make some changes to help manage things like these fishes that depend on wetlands. So summary thoughts. Native species still hold top predatory roles here in the river. We got to make sure that continues. Invasive organisms create uncertainty here. So does climate change, the environment, changes to our habitat, development, and all these organizations that you support are part of that, maintaining the line. Hydrology is a key to habitat. If you can have habitat, if you can't put water on it, it's not going to be any good to these fish. And it's way more complex than that, like nutrient dynamics, farm runoff, all these things are important to maintaining quality habitat. So we're all about protection, restoration, and management. More and more people are going to come love the river, so we got to be ready for it. And here's how you can help. My shameless pitch for the Tibbs Friends program to help support us. And thank you for your time. I don't know where I am on the schedule, but I hope you enjoyed that. Bridget, do I have time to take any questions? I'm feeling like blinded.
If somebody have any questions for Joey, we can take a couple.
I can. Yes, please.
So I want to thank you for not following some of your colleagues without this right here in God's country. A question for you. Acid deposition. Have you started, is that part of your study? Have you seen anything
Could you repeat that last part?
for the acid deposition?
Oh, acid rain? Yeah. So in the St. Lawrence watershed, which extends up into the Adirondacks, we currently actually have a study where we're comparing Cranberry Lake to the river, because the lake has introduced pike, and they have native plants, and the river has native pike and non-native plants. So I have a sea grant project right now with my colleague, Dr. Emily Arsenal, like studying the watershed properties. But the point being is, you mentioned granite, so those Precambrian gneiss, not the I'm being nice, but the GNEISS gneiss, they don't have a lot of acid neutralizing capacity, but we're here in the Frontenac Arch is similar, that it has that type of rock, but the soils here are primarily limestone underlied, and we have thicker subsoils, and they're able to buffer acid rain. So when we measure pH in the St. Lawrence River, it's alkaline. It's near 8 on the pH scale, whereas in the Adirondacks, you'll see the acid rain effects, which have gotten a lot better. Brook trout are starting to reproduce again in the Adirondacks because of, so when people talk about we can't do anything about climate change, we've solved some problems before that were pretty significant. And not that acid rain is going away, but it's reached a point where fish are starting to reproduce again, which is pretty exciting. So thanks for that question. Anything else?
Have you found that the chain or the grass muskie are susceptible to virus?
So the chain pickerel and right. So that's a really, so the virus is healthy and evolving, with the round goby as being the primary vector. And interestingly, northern pike have not had a similar die-off, despite them being what we call congeners, which they share a genus, so they're very similar, like phylogenetically, so they're really similar fish, they're cousins. But northern pike haven't had die-offs like we've seen in muskie. Muskie are sensitive. Chain pickerel seem to be performing incredibly in the river. They're going to get more abundant. So we haven't seen any VHSV effects on those fishes. Yep. Yes?
Farrell, I was wondering if you do any evaluations after the channeling and top pulling, how many years down the road to evaluate kind of long-term how they function?
Yeah, like in terms of them replugging up with typha and things of that nature. Yeah, it's I was really skeptical of doing this in wetlands. Some of the projects are focused on recreating historic connections. Other projects were focused on reconnecting the main creek channel across the zonation to reach the wet meadow. There were projects done by DEC in the 50s where the channels are still intact. So, when you dig into the substrate, we're not seeing them fill in per se, but what we do see in some cases is the floating cattail mats will break off and float into them and block them that way. So that is a process that's occurring that hasn't like ruined all the projects, but some percentage of the connectivity was lost, due to that problem. I think it would be called restenosis in the cardiology world. So anybody else? I feel like I've taken too much time, but all right, well, thank you so much.